My transfer to Io Agricultural Research Colony felt like both a massive leap and a return to roots. A leap, because Io, one of Jupiter's harsh, volcanic moons, terraformed into a fragile bubble of breathable atmosphere and arable soil, was about as far from Earth's verdant fields as you could get. A return, because I was stepping into the shoes of Dr. Theo Wells, my former professor, mentor, and the man who had instilled in me a deep fascination with exobotanical adaptation. He had retired from his colony post to lead a new scientific board back on Earth, and his recommendation had landed me this job. Big shoes to fill. The colony itself was functional. Domes of reinforced transparent steel covered vast tracts of artificial soil, filtering the perpetual, eerie twilight of Jupiter's shadow. Inside, rows upon rows of genetically modified crops thrived, strains designed to withstand the unique soil composition, the altered gravity, the filtered light. My predecessor, Wells, had been instrumental in developing many of these variants. His legacy was literally rooted in this soil. My first month was a blur of inductions, familiarizing myself with the colony's systems, the peculiarities of Io's environment, and the rhythms of the research cycles. The team was competent, if a little, set in their ways. They'd been working under Wells for years, and the routines were well established. I settled into my new office, sterile, efficient, with a single window offering a breathtaking, and slightly terrifying, view of Jupiter's swirling, gaseous surface hanging immense in the sky. The desk was standard issue, cleared of personal effects, ready for its new occupant. It took about a month, once I was through with the initial handover and began focusing on monitoring crop performance data, that I started noticing it. It wasn't in the headline reports. Those usually focused on overall yield percentages, energy consumption, pest resistance metrics. What I saw were subtle, localized inconsistencies in the detailed yield analyses for specific sectors. Certain plots, specifically those growing Wells' later-stage research variants, the ones with enhanced nutrient uptake, showed a fractional increase in yield. We're talking percentages just outside the expected deviation range, but not enough to trigger an anomaly flag in the automated reporting system. Just slightly better. My initial thought was sensor error. I recalibrated the yield monitors in those sectors. The next cycle, the data came back the same. Slightly, inexplicably higher yield in those specific areas. I checked the soil composition reports for those plots. Standard Io artificial substrate. Uniform nutrient mix. Trace elements were within expected ranges, maybe a tiny bit elevated in certain compounds, but nothing statistically significant, nothing that explained the consistent yield bump. I talked to the other agriculturalists. Hey, have you noticed anything weird about Sector Gamma 7? Old man Hemlock, who'd been here since the domes went up, just shrugged his shoulders, the movement stiff in his environment suit. Gamma 7? Good crop, that variant. Wells' best work, maybe. Io soil's got its moods, kid. Don't chase ghosts in the data. His response was typical. They saw the slightly better yield as just. Io being Io. Or Wells' genius manifesting in unexpected ways. No curiosity about the why. The official reports from the past few cycles were equally unhelpful. Pages and pages of data, logs, performance metrics. No mention of localized yield increases outside the norm. It was like the anomaly was too small for their systems to care about, or for their busy minds to notice. The mystery, small as it was, bugged me. Why those plots? Why that variant? It felt like a puzzle, a quiet little secret the colony was keeping. One afternoon, rummaging through the drawers of my desk, looking for some old project files Wells might have left behind, my hand brushed against something tucked away at the very back of the bottom drawer. A small, worn, leather-bound notebook with Wells' handwriting. This wasn't an official log. This was personal. A diary, a scratch pad for his thoughts, observations that didn't fit neatly into formal reports. His neat, dense script filled the pages, interspersed with quick sketches and complex equations. It was a window into his mind, the mind that had helped build this colony's foundation. I flipped through it, fascinated. Notes on early terraforming challenges, observations about stellar radiation effects on plant growth, philosophical musings about humanity's place among the stars. And then, I found it. A series of entries over a few weeks, dated roughly 6 months before his departure. Notes on monitoring the same crop variants in the same sectors I'd been looking at. Entries like, Cycle 237. Yield data for variant 9b and gamma 7 shows slight positive deviation, plus 0.004%. Recalibrated monitors. Likely sensor drift. Cycle 240. Variant 9b continues showing elevated yield, plus 0.006%. Also noted in Delta 2, variant 11c. Soil analysis inconclusive. Cycle 245. Yield anomaly persists, plus 0.009% in gamma 7. Reviewed environment logs, no correlating factors. Curious. He'd seen it too. The small, nagging anomaly that the official reports ignored. He'd checked the obvious things, just as I had, and found nothing. My excitement mounted. I wasn't chasing ghosts. Wells had seen it. He'd marked it. And then, on one of the last entries related to the anomaly, a short note, almost tucked into the margin. Observed microscat, but no indication of source. Microscat. Tiny droppings. But no indication of a source? He'd seen droppings and suspected they were linked? But why hadn't he investigated further? His promotion. His move to Earth. He must have been too busy, too focused on the transition, to chase down something so minor, so strange. He'd left the thread hanging, a mystery noted only in his private journal. A footnote. That footnote became my mission. Observed microscat, but no indication of source. What was the source? I went back to the detailed soil analysis reports for the anomalous sectors. I didn't look for chemical composition anymore. I looked for microscopic foreign biological material. And yes, buried deep in the raw data logs, often filtered out as environmental particulate, were mentions of minute traces of processed organic matter. Microscat, just as Wells had noted. But scat from what? Nothing officially lived outside the domes. Pests were contained. Microbes were cataloged. I needed eyes on the fields themselves, eyes that could see more than the automated monitors. I requested and installed a series of low-impact wildlife observation cameras in the sectors showing the anomaly. Just basic models, used for monitoring fungal growth patterns or insect activity. I angled them low, towards the soil, hoping to catch sight of whatever was leaving the microscat. Weeks passed. The cameras recorded the slow, deliberate growth of the modified crops, the automated irrigation systems, the occasional scuttling insect. And then, finally, reviewing the footage from Gamma 7, I saw them. Tiny shapes, darting among the base of the plants. Hard to make out at first. They were so small, moving so fast. But as they paused, caught in the camera's low light, their forms became clearer. Small, furry, but with delicate, almost translucent wings folded tight against their backs. Faces that were a strange mix of rodent and something else, something altered. They moved with a quick, scuttling gait, occasionally stopping to nose at the base of the plants, sometimes eating something small near the roots, then leaving behind the microscat Wells had noted. I watched them mesmerized. They seemed particularly attracted to the variant 9b crops in that section. They weren't pests. They weren't damaging the plants. They were interacting. I knew I had to get a closer look. Carefully, over several nights, I set up humane live traps, small, baited enclosures designed for collecting insect samples. It took patience, but eventually, I captured a few specimens. Holding one in a containment field in my lab was surreal. It was a lot smaller than my hand, soft-furred along its body, with those delicate, non-functional wings and a twitching snout. A quick genetic scan confirmed my initial thought that they were a type of glires, a lineage of subterranean mammals that had been discovered in Io's lava tubes before terraforming began. But their current form? This wasn't the burrowing, standard glires on the colony's original biodata surveys. This was a dramatic mutation, a significant divergence from the baseline species, somehow triggered and accelerated by the unique environmental pressures and biological factors of the terraforming process itself. A new, uncatalogued variant, living right under our noses in the artificial ecosystem we had created. My searches of the colony's biodata logs turned up nothing on this specific, mutated lineage. The original glires were noted, their biology studied, but these small, winged, surface-dwelling versions? They were living footnotes of the terraforming, overlooked in the focus on creating viable plant life. The scat analysis came next. Microscopic, chemical, genetic. This is where Wells' cryptic note truly paid off. The scat contained a complex mix of enzymes and processed organic matter, unique to these mutated mammals. And when I cross-referenced its composition with the specific nutrient requirements of the variant 9b and variant 11c crops, it was like finding a perfectly tailored key for a complex lock. These specific crop variants, engineered by Wells for enhanced nutrient uptake in Io's soil, had inadvertently created a perfect food source for the mutated mammals. And the mammals, eating these specific plants, were producing excrement that acted as a hyper-efficient, perfectly balanced fertilizer for those exact same plants. A symbiotic loop, created by chance and mutation, playing out in the fields, boosting yields in a way no one had understood. No indication of source in Wells' note wasn't just about the scat. It was about the mammals he'd glimpsed, the source of the scat, the missing piece he hadn't had time to identify. My research project shifted dramatically. Forget the minor recalibrations and yield monitoring. I had found something far bigger. The potential implications were immense. Could this biological interaction be replicated? Could we create a natural, self-sustaining fertilizer system using engineered crops and specific, symbiotic organisms? Not just on Io, but on other colonies, other worlds? My colleagues still saw the anomaly as Io being Io. The official reports remained oblivious. My discovery, for now, was confined to my lab, my data logs, and the echo of Wells' cryptic footnote in the bottom drawer of my desk. It wasn't a galactic war or a diplomatic incident. It was a quiet, scientific mystery solved in a dome on a moon billions of kilometers from Earth. A new, mutated species, a unique biological interaction, a potential revolution in exobotanical agriculture, all stemming from a tiny increase in yield and a handwritten note about microscat. My work had just begun. The story of the little mammals and their tailored fertilizer was nowhere near the main text of galactic scientific journals yet. But I was writing the first chapter. The most important discoveries I was learning often started as the smallest, most overlooked footnotes. And I was ready to see where this one led.